אנחנו בהרכב כזה חלקי היברידי לאור התקופה, כשלמעשה פרופסור סצ'י-פאינרו זכתה בפרס, היה אמור להתקיים טקס בשנה שעברה בזמן הזה, בדיוק באמצע הסגר הראשון, והחלטנו לדחות את זה. וגם כשקבענו את המתכונת הנוכחית לא ידענו אם אפשר יהיה לעשות את זה במתכונת רחבה או לא, ולכן החלטנו על מתכונת היברידית שעכשיו כנראה הוכיחה את עצמה עם כל האירועים האחרונים. אז אנחנו מודים לכל מי שהצטרף פיזית ולכל מי שמצטרף היברידית לכנס, ושמחים באמת לארח כאן את פרופסור רונית סצ'י-פאינרו, שזוכה השנה בפרס משפחת יודעים למצוינות בחקר הסרטן, וכמובן מברכים גם את פרופסור יודעים מהמשפחה שמכבדים אותנו בנוכחותם. אני, לפני שנתמקד בפרס הנוכחי, רק אזכיר שבשנה שעברה לא היה קול קורא לפרס של השנה הנוכחית, אבל הוא מתחדש עבור השנה הקרובה, ויש לנו חידוש השנה של שני פרסים מעבר לפרס החוקר הראשי, שמיועדים לתלמידי דוקטורט, תלמידים בשלבים מתקדמים ל-PhD, שגם הם יוכלו להגיש מועמדות, ובאתר אתם תוכלו למצוא את כל הפרטים לגבי הפרסים האלה. הפרס לשנת 2019 או 20, לפרס יודעים למצוינות בחקר הסרטן, מוענק לפרופסור רונית סצ'י-פאינרו, על המחקר הבינלאומי פורץ הדרך שלה, שקשור גילוי של מנגנונים מולקולריים בכל מה שקשור לאינטראקציות בין המאחסן לגידול. ובאמת המחקר הבאמת פורץ דרך הזה הביא למספר גדול גם של פרסומים בעיתונות המובילה ביותר, וגם לפיתוח של אפשרויות טיפול, גישות טיפוליות שנשמע עליהן היום, לטיפול בממאירויות שונות. אז אני רוצה להזמין קודם כל לשלב הברכות את דוקטור מיקי אלברטל, מנהל בית החולים רמב"ם.
שלום לכולם, פומה ופרופסור יודעים, we made it at the end, you know, we postponed and again and again and again, but it's done. Professor Biar, Professor Sacchi-Fainaro, oh, I see that the family has arrived. So, למעשה אנחנו מדברים על, זה כבר טרנדישן, כן, מ-2013, של מתן פרס של פרופסור יודעים למצוינות ומחקר בסרטן. רשימת הזוכים היא מאוד מרשימה, זה מתחיל מפרופסור אלי פיקרסקי, שאני חושב שהיה הראשון, אחר כך יובל שקד, בני דקל, ירדנה סמואל, סידו וולף, רמי אקילן וזיו גיל, והזוכה השנה, מזל טוב, זה לא שנה שעברה ולפני שנתיים, כן? אבל פרופסור רונית סצ'י-פאינרו, ואנחנו שמחים שהצלחנו לדחוף את זה לפני שאולי הקורונה משפיעה עלינו, למרות שאני חושב שבסך הכל זה יהיה בשליטה. צריכים גם להזכיר, ולא יצא לי להביע אישית, זה להשתתף בצער של המשפחה, בפטירה של אחיך, המפרי, וגם כן הזדמנות להביע רגשות השתתפות בצער. הקידום של מחקר בבית חולים רמב"ם, ואנחנו אומרים את זה בכל פורום, הוא ברמה של אם לא נהיה שם ולא נקדם את המחקר ולא נקדם את החדשנות, לא נהיה רלוונטי. כלומר, אם אנחנו באמת, כמו שמופיע בחזון של בית החולים, שאנחנו רוצים להיות מרכז קליני מוביל, המעורבות של מחקר וחדשנות חייב להיות באותה דרגת חשיבות, והעובדה שיש אמצעים כמו הפרס הנוכחי שגורם באמת להוביל מחקרים הוא נושא מאוד משמעותי, ולכן הערכתנו הגדולה מאוד לך על הפרס הזה. ואנחנו באמת רואים את רשימת האנשים שכבר קיבלו את הפרס, באמת, האנשים המובילים בתוך המערכת, ולכן החשיבות שלו. אז אנחנו קודם כל מאחלים שיהיה מפגש נעים, ותודה שוב לכם, ובהצלחה, ונשמע עוד דברים. אז תודה רבה.
אני רוצה להזמין את פרופסור ביאר.
תודה, פרופסור רונית בן אהרון, דוקטור אלברטל, פרופסור יודעים and פורמה and the family, המשפחה, חברים נכבדים, א', תענוג לי להיות פה שוב יחד איתכם בחדר הזה, בקומה השביעית של מכון פישמן לאונקולוגיה, עם הצוות הכל כך מסור ונאמן, ועם הזוכה שלנו, פרופסור פייינרו מאוניברסיטת תל אביב, ברכות על הפרס הזה. אני חושב שזה 7 שנים כבר, או 7 מפגשים, זה כנס השביעי שאנחנו נותנים את פרס יודעים, וללא שום ספק זה פרס שעשה אימפקט בישראל, אנשים מתחרים עליו, זה כבוד גדול לקבל את הפרס הזה, זה מוכיח את העשייה בתחום הסרטן במדינת ישראל, ואני חושב שאנחנו יכולים להתברך בקידום מאוד, הייתי אומר, מאוד מהיר של מחקר הסרטן בישראל, ולשמחתי הרבה, גם בית חולים רמב"ם הוא חלק מרכזי מהקידום הזה. קודם כל, עם הקליטה של פרופסור רונית בן אהרון כאן, אני חושב שהמכון כולו קיבל דחיפה אדירה קדימה, ואני מברך אותך שוב פעם, הזדמנות, 2 לא הייתי פה על הבמה הזאת, אז זו הזדמנות. דבר שני, אני חושב שהיכולת כאן בבית חולים רמב"ם, בין המחקר של רמב"ם בסרטן, המחקר הקליני, בין הפעילויות שקורות בטכניון בבריאות האדם, בין ההקמה של מרכז למחקר הסרטן על שם רפופורט, כאן לידינו, בראשות פרופסור צ'חנובר, וכל השותפים מהטכניון, יחד עם שותפים בינלאומיים, ממוריאל סלון קטרינג וסטנפורד, הכל בתחום הסרטן, נותנים כאן קר, גם לפעילות מאוד נרחבת בין הרפואה והאקדמיה, כאן בבית חולים, וגם בכל העולם, דרך השותפים שלנו. ואני חושב שדוקטור אלברטל מאוד פעיל בכל הקידום הזה של שיתופי הפעולה הבינלאומיים, וגם בתוך הטכניון, ואני מאוד מברך אתכם על הפעילות הזאת. אני חושב שיצאו מכאן עוד הרבה מאוד תובנות ומחקרים וטיפולים חדשים למחלות הסרטן כאן שלפנינו. אז אני לא רוצה להאריך, אני שוב פעם מודה לכולם, מודה במיוחד למשפחת יודעים על התרומה רבת השנים ועל הדחיפה של מחקר הסרטן בישראל, וברכות לזוכה, ותודה רבה לכולכם ובהצלחה.
I would like to invite you, Professor יודעים.
Thank you very much, Rita. I just want to tell you why I made this prize. Because a significant number of my really close family and my wife's family died from cancer. And 6 months ago, my brother died in Los Angeles. And most of that work they've done in neurodegenerative diseases like Parkinson, Alzheimer, I really learned from going to cancer meetings, because many of the mechanisms that are involved in neurodegeneration are completely opposite to that of cancer, the molecular mechanism. Amazing. And some of the drugs I developed was to develop the opposite to what they're doing in cancer. I'm really delighted that Rna is receiving this prize. This morning I was really reading your paper and I listened to your video, and immunotherapy seems to be everywhere, in Parkinson, Alzheimer, and cancer. But unfortunately, in Alzheimer, I know there are 2 new drugs. There's tremendous exception by the experts in the field. It really is not a cure for Alzheimer, it's just getting the plaques out of the brain. I also want to especially thank Rafi. I know Rafi for a long, long time. When he was in biomedical engineering, I was proud to be sit on the committee when we made him a professor there. I remember beside him. And when he back came to the faculty, the same, and he became professor in faculty. And so we have worked together. I'm delighted that he accepted to develop this prize. We have got some of the most outstanding people. You are one of them, really. And to me, this is really very special that we've got some wonderful, brilliant people that are working in Israel. I want to thank Neta. Who is Neta? She really organizes the meeting. Neta, thank you very much. I want to the whole committee, and especially Professor Cotton. He can't be here, but he's on Zoom, and he's really a wonderful friend of ours. So, Rita, I wish you all the success. I hope we can develop the drug with this new protein, that we can break the back of this protein, maybe with monoclonal antibody, new antibody. So I thank everybody that's here. We're looking forward to your presentation. Thank you very much.
Thank you. פרופסור רונית סצ'י-פאינרו תיתן לנו עכשיו איזושהי טעימה מאחד מיני רבים ממחקרי פורצי הדרך, ואני באמת גאה שאת כאן איתנו, וברכות על הפרס.
So, good afternoon, everyone, and I would like to thank Professor יודעים and his family for this amazing commitment to cancer research and the translation from basic research to the clinical side. I would like to thank Professor Ben-Aaron, Professor Abetal, Professor Biar, Professor Cotton, who I understand is on Zoom, and of course to Neta for organizing this really prestigious award and ceremony. And I feel humbled to get this prize, and I thank you for here for getting this award. So thank you very much for giving me the honor to stand here and tell you about my research today. So I will share with you one of the projects running in my lab. This project is an example to many other, to the way we research cancer, taking it from the bed to bench and back to the bedside. And the example that I will give today is actually from an unpublished work on melanoma brain metastasis, but it has a common denominator with other projects in the lab where we research the interactions between the tumor and the host, mainly focusing on the brain in this part of the work. And I'll share with you what was the origin for this work. So about 30 years ago, in 1993, there was a study published at the New England Journal of Medicine, summarizing the results and the findings from 16,000 autopsies of people that died of trauma. These were people that died of car accidents, of suicide, and other reasons, definitely not for cancer or any other known disease. And what struck me as fascinating in a way, if we put aside the emotional part, but the interest of science and the clinical part, is that about 46% of the men in this study from the autopsies given had these tiny in situ carcinomas in their prostate. 39% of the women had these tiny, again, in situ, meaning inside lesions in their breast. And virtually all, 99%, had this tiny avascular, when I say tiny, is about 1 to 3 millimeters in size, of thyroid in situ carcinomas. So this is really depressing, and this is not why I came here to tell you today, but rather from the side of the clinical. Meaning that if we compare it to how many are clinically diagnosed, meaning people that came with symptoms arriving to the physician, only 1% in this age range for prostate or for breast in men and women accordingly, and 0.1% for thyroid carcinomas. So that actually gave me some light at the end of the tunnel, saying that maybe we should look not on why people get cancer, but rather why not. Maybe we should look into the mechanisms of prevention. How come our body makes sure that these tiny, what we call dormant or occult lesions, stay dormant for probably the rest of our lives until the age of 120 without even knowing that they are there. And what happens in those that do switch on and get out of dormancy. So I'll give you the example of brain metastasis today. And the reason I started this research was about 6 years ago, in 2015, I heard a fascinating talk at MD Anderson, where one of the dermato-oncologists mentioned that 90% of advanced melanoma patients present occult metastasis in the brain. And I was so disturbed by that at the end of her talk, I came and asked, did I miss it? Is it 90 or 19? Maybe I didn't hear well. She said, no, 90. And if you think about it, this is not trivial. I mean, if she would have said the same on lung metastasis or liver metastasis, that would have made sense. But brain, we have the blood-brain barrier. It's a protective niche. It's not supposed to be a hospitable environment for cancer cells, for circulating cancer cells. So we started researching that, and our hypothesis was that if we look at the primary lesion, and we're doing the same research that is sponsored by the ERC Advanced Grant on melanoma, breast cancer, and lung cancer brain metastasis. So if we take the example of melanoma, when we look at the primary site, so we have some cells that leave the original primary site, they intravasate into the blood vessels, they start circulating, and while in circulation, they secrete cytokines, chemokines, different proteins that are pro-inflammatory, and it all depends on who is listening. This is the way cells communicate. Who is listening? Who is taking up on these signals from the cytokines? I have 3 boys in my house, and when I see them with their mobile phones watching the screens all the time, I tell them, conversation is when I speak and you answer. It's not only one side. So here we wanted to see who is listening and who is responding. So it could be that there are cells responding in the lung and cells responding in the liver, but there were cells responding in the brain, and those cells are the microglia and the astrocytes. And what we hypothesize is that if they respond, that means that they will become activated and secrete other pro-inflammatory cytokines in return. And indeed, for this step, we took about 100 sample FFPE, formalin fixed paraffin embedded tissues, and we will stained for activation. And we saw that in all those cases, we see activation of microglia, looking at IBA1 staining, and GFAP for astrocytes activation. That led to the secretion of cytokines, a reciprocal secretion coming now from the activated residents in the brain that secrete other factors. And those pro-inflammatory factors cause neuroinflammation that induce astrogliosis and microgliosis, meaning now astrocytes and microglia will start dividing and proliferating, and also stimulate neuroinflammation that will lead to angiogenesis, the recruitment of new blood vessels from pre-existing ones, and hyperpermeable vasculature. So the blood-brain barrier is no longer intact, but rather it is leaky, and now it can allow circulating, in this case melanoma cells, to extravasate to the brain tissue, to this homing effect to the neuroinflammation, and then create those occult, dormant, in situ lesions that will slowly, with time, slowly or more rapidly, will start recruiting blood vessels, angiogenic blood vessels, and help in the growing of this mass in the brain until the patient succumb to the disease. So this was our hypothesis, and the way to tackle it was first to create models. We are mouse doctors, so we first create spontaneous brain metastasis models, meaning that we inoculate the primary lesion intradermally. In the case of melanoma, again, we do the same for lung, for intramammary, for breast cancer, to initiate a spontaneous development of an establishment of brain metastasis. We wait for a while, then we excise the primary lesion and wait. We wait until we see by non-invasive intravital imaging modalities, MRI, CT, or fluorescence detection, we see when we get these brain lesions. We do it over and over again. This is also in collaboration with Professor Rna. We did it on melanoma and now on breast cancer and other colorectal carcinomas. And we see that slowly with time, we go back and forth from the brain to the orthotopic site of the primary lesion, and we get them more and more metastatic. Then we use all the possible omics, meaning we do RNA-seq, single-cell RNA-seq, proteomics, whole exome sequencing, and other omics. And we look at mutation sequencing under the light, but also a fishing expedition to see what is different between the primary and the metastatic site in terms of the tumor progression, to see what is different. And when I say different, I mean any deregulation, whether it goes up or down in its regulation of those cytokines. And then when we get several candidates, we select the ones that can be druggable, meaning that if we block them, if it is overexpressed, or if we express them, if there is low expression in those metastases, then we see that they are driver mutations, meaning that they will lead to increased proliferation, migration, invasion, and metastatic phenotype of the tumor. So if we block them, we want to see that the tumor is not progressing. So we had several of those candidates, and then sometimes we are lucky, and there is already a drug that targets the same protein, the same cytokine. We were lucky with that in our latest research that was published on glioblastoma. This is what Professor יודעים referred to on P-selectin. So sometimes there is for a different indication, so we can easily, more or less, repurpose it for our target. So we can take those drugs and combine them as a cocktail that anyhow, all oncology fields we use these cocktails, but in this case, we put them in one nanoparticle. So not only the drugs are injected simultaneously, but as you all know, they have different PK, different PD, and having different half-lives, they do not reach the tumor at the same size. So using nanotechnology, we can make sure that they are delivered and activated and released at the tumor tissue selectively, then exerting their synergistic activity, and we can see the combination index really taking place when they arrive simultaneously to the tumor, and not necessarily when they are injected together, but one has, in this case, for example, with the BRAF and MEK inhibitors, as we know that BRAF V600E is a mutation of 50% of melanoma patients. So in this case, when we inject them, as approved by the FDA, at the same time, one has few minutes half-life and the other has 24 hours. So they definitely do not reach at the same time to the tumor. So this is one option. And the other, if we are not that lucky and we do not have a drug that is already out there and approved even for a different indication, then we can use Rna Therapeutics. I'm sure that after the last year, we all know about Rna Therapeutics. So we either encapsulate in our nanoparticle mRNA if it's a missing protein, or use CRISPR or siRNA to silence a gene if we want to block it. So these are our options. And of course, neutralizing antibodies, which became a routine in clinical oncology. Then we test them on the melanoma cells, in this case, that are labeled with mCherry, as you can see here in the dish. We move to in vivo, to mice, where we inoculate, and we see the tumor here non-invasively, intravitally, that is in the mouse brain as a melanoma brain metastasis, and then hopefully move to patients. This is a long process, as you know. It takes 5 to 10 years if everything goes well. So we start with the models, and as you can see here, we managed to get a spontaneous metastasis in the brain, as can be seen here by MRI. See that we have a whole array of different melanoma models from patients and murine from mouse, because we are also interested in immunocompetent mice as we are looking at immunotherapy. So it's important to have immunocompetent mice that have T cells and B cells in this case, and definitely dendritic cells that start the whole process. And 50% have the melanoma, the BRAF V600E mutation, some other mutations in the BRAF gene, and some are wild type. So we can cover all the different clinical options. And as you can see here, looking at my original hypothesis, so we can see the D4M is a BRAF mutated murine melanoma. The 5B1 is the melanoma metastasis from patients. And here we have plenty of samples from melanoma brain metastasis from patients directly taken to the lab. In all of them, we see in green here IBA1 expression, meaning that the microglia are indeed activated. Here the GFAP staining in green, again in all three we can see that the astrocytes are activated, and CD31 showing angiogenesis, the recruitment of new blood vessels and higher microvessel density. Not only that, we see that those vessels are leaky, proving our hypothesis of the neuroinflammation leading to the compromise of the blood-brain barrier, as can be seen here by Evans blue. And in MRI, we see the leaky vessels in those lesions in the brain, as opposed to the rest of the brain where we have intact blood-brain barrier. So back to our platform and the way we process our samples, we had about 5 years ago neurosurgeons joining our lab, and they offered very kindly to come and to their lab, meaning the operating suite, and start from the patients. So now we did everything in reverse. We started from the patients, we came to the surgery room, take a chunk, implant it in the brain of the mice, and do everything in reverse. That was much faster. It takes about 6 to 9 months until we have enough mice and colonies to do our research on them. But that's too long. If you think about it as a clinician or as a patient, 6 to 9 months for melanoma brain metastasis, it's a really long time. Either the patient already succumbed to the disease, or the tumor that I got 9 months ago has nothing to do with the tumor that patient has after 9 months. It already mutated. Those are selective challenging of the cells that remain there. So we got the professorship, a lot of papers, but is it really helpful to the patients? So we had to think out of the box. And for that, we had Lena Neufeld, the chemical engineer, coming to the lab, Noah Reisman, an architect, and Yael Stillerman, an MD PhD. And the way we thought about it was to start from the notification that there is a patient coming that has a brain metastasis, then get the MRI prior to the surgery, do the image analysis, convert the file to STL file that a 3D printer can read. And now on the day of the surgery, we come to the surgery room, we take a chunk, we create cell suspension, but only now we have all the cell populations, meaning not only the cancer cells that we usually seed on 2D rigid plastic, but also the astrocytes, the microglia, the endothelial cells, the pericytes, all of them together. And we connect the blood vessels that are created, we connect them to a microfluidic system that we can flow the PBMCs of that same patient, making it relevant to immunotherapy. Because when we take a chunk from a patient and implant it in an immunodeficient mouse, the mouse doesn't have a proper immune system, so we cannot test immunotherapies. So what we did here, this is a process that takes only 2 weeks, and we can test on it on about 100 mini-me tumors of the same patient. And we, as a polymer chemist originally, we create hydrogels from polymers that are biodegradable, and we fit all the mechanical properties of the tissue, the extracellular matrix, to lung, to brain, to breast, and so on, because we cannot compare how only tumor cells grow on rigid plastic and then be surprised that nothing is translated to the clinic at the end of the clinical trials. We cannot be surprised from 95% attrition rate when we are not mimicking the actual clinical scenario. So what we are doing here, this is how it looks from outside. This is the brain tissue that has all the cells from that patient, but inside you can see from the MRI translation, the image analysis to the STL file in the 3D printer, you can see that we printed the blood vessels. Here you can see that they create a life of their own while creating new blood vessels and branching and sprouting out. And those blood vessels have lumen. So I'm saying it as it is nothing. This sentence took about 3.5 years just to have vessels that do not collapse by the weight of the gel, the hydrogel, the tissue. And then you can see here in red are the endothelial cells. In light blue are the pericytes that stabilize the blood vessels, the cancer cells in blue, the astrocytes in green, and microglia unlabeled. And you can see all the cellular components here in this tissue. And the blood vessels are functional, so we can flow through them the therapies that we are testing or the PBMCs of the patient. And you can see here in this slide from the 3D, let's call it an organoid, the tumor organoid sort of, we can test and predict by that if the therapy, in the case of immunotherapy, if it was useful and efficacious in the model, can we predict that it will work on patients? You can see here in those that it worked, we can see by staining CD8 T cells, we can see the infiltration here into the tumor, the 3D bioprinted model. And moreover, when we isolate the cells, if we look at the tumor cells, they create, imagine this is a 3-dimension tumor. You can see it here growing in mass. And here on the side, when we take also, if it is splenocytes from the mice that were treated with the immunotherapy or from the PBMCs that were activated, we can see how they destroyed the tumor cells here in red. We can also follow up the penetration of the drugs if we label them with a different like Cy5 labeling probes. So back to our model, if we look at all the different sites, we would like to intervene either in the primary, right after the excision for prevention using a dendritic cell targeted nanovaccine, or intervene while silencing important or driver cytokines that are overexpressed, or in regression when usually patients come already when they present the metastasis, targeting the tumor cells directly with BRAF MEK inhibitors that are encapsulated in one single nanoparticle together. But before that, we have the surgery cited. As my title also mentioned excision prior to diagnosis and treatment. You can see very quickly here how we developed a probe that is a turn-on probe for image-guided surgery, that at the time of the surgery, if we inject prior to the surgery, about 1 hour before, we inject our probe that is a polymeric nanoparticle that is chewed up by cathepsins that are overexpressed in the tumor tissue. They arrive to the tumor site due to the enhanced permeability and retention effect. Those same leaky vessels that I mentioned before, it extravasates from the leaky vessels, internalizes by endocytosis to the tumor cells, and is being activated, so it turns on its fluorescence only at the inside the tumor cells and nowhere else. So at the time of at real time during the surgery, the surgeon can see that they are shine light, seeing exactly what are the margins of the lesion. So this was our first attempt to try to prevent the metastatic cascade. So making sure that we are not taking too much of the tissue, but not too little either. So then we move to a vaccine for prevention. So following the surgery, we start with a targeted nanovaccine, where you can see here a nanoparticle that is made of a degradable polymer that is decorated by mannose, a sugar, to trick the dendritic cells. So the dendritic cells, once we inject our nanoparticle, the dendritic cells come to the site and they have the mannose receptor, CD206, as you can see here, and they internalize the nanoparticle due to their mannose that is decorating it, and then they process either the protein that is inside. In this case, for melanoma, we use the MART1, the melanoma antigen, that this is what the pathologists use in order to define that this is a melanoma tissue. And it is, while internalizing and processing the peptides, we have inside two peptides, MHC1 and MHC2, that while in transit, the dendritic cells mature and present the MHC1 and MHC2 complexes while they reach the lymph node. And then CD8 T cells and CD4 T cells bind via their TCRs, the T cell receptor, they bind to the complex CD8 to the MHC1 complex and CD4 to the CD4 complex. And again, thanks to the, if we can find something good about the corona in the last year, is that we all learned a lot about immunology. We all became experts. So now if we always thought and assumed that for cancer, the cellular immunity is what is important, and for infectious diseases is the humoral immunity, now we know that for both it is important. And the reason we know that for both also are relevant here for cancer is that we converted this nanoparticle, like everyone else, from cancer to coronavirus research, and we replaced the MART1 antigen with the spike antigen. So we also developed a sort of nanoparticle for the COVID-19. But then we looked for neutralizing antibodies. So not only that they will be activated CD4 T cells that will activate B cells that will secrete the antibodies that are low in number but very high affinity that will target the antigen. Then by ADCC, we saw that NK cells are coming to the site, secreting interferon gamma and other cytokines and granzyme B and perforin, destroying the tumor cells. And also the CD8 that come and bind via their TCR that were activated in the lymph nodes. Now they bind to the MHC1 complex on the tumor cells, destroying them as well. So if in the past we separated those two immunities, now we know how important it is for memory in cancer. So what we saw a few years ago, 3 years ago, that in some of our studies that we waited enough, we didn't see recurrence of the disease, nor did we see any metastasis. The explanation was that we created memory. We were able, by raising those neutralizing antibodies, to create memory from the activated B cells. So now we are measuring both the cellular and the humoral immunity for the COVID-19 research and also for cancer. So to make a long story short, I won't dwell on the details on the physicochemical characterization, but this is our nanoparticle. It's about 200 nanometers in size, which is the perfect size for dendritic cells to come and swallow them and be activated. We see that they accumulate selectively in the lymph nodes, as seen here, by labeling them with Cy5. And the reason being is that we don't even know and do not need to know where the cancer tissue is. We are injecting them subcutaneously. They activate the dendritic cells that come to the site, and then they go and activate the T cells and B cells, and they do the rest of the job. So we are making our bodies a laboratory of its own without the need to take out the dendritic cells or T cells like in CAR-T, where we need a very expensive clean room in the hospital, then returning the cells to the patients that anyhow are very weak, and many times we get contaminations. So this makes all this redundant because we're using the machinery in the body itself. So I'm jumping to the in vivo, and we saw at this point that we get inhibition of tumor growth and extended survival, and we were really happy, and we submitted with great joy the paper to Nature Nanotechnology, and it came back saying, this is very nice, but we're not going to vaccinate all the population against melanoma. So now change the schedule of the treatment and show us intervention, not prevention. So we went back, we did an intervention study, and la depression. What we saw is no effect whatsoever. Both worked, they inhibited the cancer growth, but they were similar, whether we injected just the immunotherapy, anti-PD-1 with OX40 in this case, or the vaccine, we got the same results. And luckily, there were some outstanding students that didn't give up, and they looked at everything that they could look at in terms of the facts analysis, looking at all the different populations. And what was extremely interesting was this population here of MDSCs, the myeloid-derived suppressive cells. And what we saw was this great increase in this population in the tumor. We went back to the clinic, we collected tissues from non-responders versus responders, and what we saw was that in non-responders to anti-PD-1 therapy, there was this population, sure enough, in the tumor. MDSCs, myeloid-derived suppressive cells, were really high in number in those tissues. So we added a BTK inhibitor, brutinib, and we thought maybe we'll get better outcome if we eliminate that population. And again, we got this differential in inhibition of tumor growth and extension of survival, and taking from the NIH tetramer facility, we got the tetramer specific to show the specificity to the peptide. So this is not just a general immune response, but it's very specific to the peptides we internalized in our nanoparticle, as can be seen here. And also when we look at ELISpot, at interferon gamma, we see the increase in intracellular interferon gamma, also alluding to the fact that it's very specific. So this is about the excision of the primary tumor, prevention, and when we moved to intervention, we wanted to eliminate those factors locally. And Sabina Pozzi, a wonderful PhD student, came from Italy to my lab. She was looking at astrocytes' role in melanoma brain metastases. And as can be seen here in all the different models from 3 different patients, from 3 different murine models or human models compared to the healthy brain, you can see in all of them that GFAP in green is activated compared to the normal healthy brain. These were from 50 different brain tissues that we got from Johns Hopkins and the Lieber Institute from postmortem analysis. They sent it to us and we could stain and didn't see this activation. So we isolated the cells, astrocytes and melanoma cells, and from those lesions. And what we saw was that the astrocytes always contribute. The more astrocytes we put, we see higher rate of proliferation, higher rate of invasion, as can be seen here in our 3D models compared to the monoculture of just the melanoma cells, and in migrating, higher rate of migration when we add the astrocytes or the conditioned media from the astrocytes, which means that something that the astrocytes are secreting once they meet, remember that interaction that I mentioned in the beginning, their conversation, then if there is something in the medium that they secrete, that will affect the melanoma cells to proliferate, invade, and migrate faster. And we did for that proteome array, and to make, again, a long story short, one of the candidates that we found was MCP1, and we wanted to see if we inhibit it, do we manage to inhibit the whole process of metastasis. And indeed, we used first a neutralizing antibody, then binderit, a small molecule inhibitor. And as can be seen here by MRI, we managed to inhibit the melanoma brain metastasis in the mouse and extend overall survival. And you can see the increase of CD8 T cells here in the tissue compared to those that were not treated, and PD-1 was inhibited. So we're managing to inhibit the immunosuppressive microenvironment and also stimulate the T cells to internalize. We did also CRISPR, same happened here by inhibiting it. And when we used the really aggressive model where none of them is effective, when we combined, we got a synergistic activity, the vaccine with the binderit, we saw that inhibition of MCP1 and activation of the dendritic cells, we got a synergistic effect leading to the inhibition of this spontaneous metastasis, can be seen here by MRI. So last but not least is the regression, because at the end of the day, we see that all of them escape at the end. And this is really worrisome. We didn't manage to cure them completely. There were some incidents that we did seek cure, but this cure is a big word. And we wanted to make sure that we take the third dimension and directly affect the tumor cells themselves. And this is what we saw here. We managed by green chemistry, one-pot chemistry, very simple. We combined the BRAF inhibitor and a MEK inhibitor, the brafenib and selumetinib on one nanoparticle, polyglutamic acid, the same polymer that is activated and cleaved by cathepsin B, an enzyme that is overexpressed in the tumor cells, and also in tumor endothelial cells. And here again, you can see in the 3D model that we managed to inhibit the sprouting of the cells and inhibit tumor growth in the mouse. So again, this is a wonderful collaboration between Sabina, Dikla, and Zhenya. and Racheli Blau, who is also already doing a postdoc in San Diego, and Diana Epstein, who is finishing now her PhD. So they did show that when we put them in one nanoparticle, that they accumulate selectively in the tumor, as opposed to the same free drugs that are used in the clinic. This is the combination that is approved. Again, we see much higher accumulation in the tumor for the conjugate compared to the free drugs. And even when it is combined with anti-PD-1 and CTLA-4, the combination that is approved for brain metastasis, and when this is in orange here, and even when we combine it with BRAF/MEK inhibition, again, the protocol used in the clinic, see how much we can prevent the brain metastasis when we use the combination with when they are inside the nanoparticle. So to summarize, this is how we approach taking it from bedside to bench and back to bedside, hopefully. This is done with this wonderful group of people coming from biology, medicine, engineering, and chemistry in the lab, and our wonderful collaborators, Adi Barzel's lab working on the CRISPR, Elena Florindo working on the vaccine, and of course the funding agencies. And just to summarize this bioconvergence approach, Judah Folkman already said that when you drain the water from the ocean, you'll find out that all the islands are connected. So this is what I'm here for, connecting between the basic science to you, to the clinicians. So thank you again, Professor Musa Yadim and the family and all the other colleagues. Yes?
You showed that melanoma is outside the brain, and yet the microglia has been activated, become amoeboid. So what is causing the microglia become activated?
No, microglia is activated prior to when the melanoma cells are circulating, they are already secreting those factors that some of them reach the brain and they activate the microglia and the astrocytes. They in return secrete factors that home the circulating cells to enter.
This is exactly what happens in Parkinson's and Alzheimer's, because we get the cytokines, inflammatory process, and yet you would think that the cytokines should actually kill the melanoma. Why are they doing this?
So this is the other project that I mentioned.
This is already finished, and we know that when we start working on progressive inflammation, you know how much money was spent developing anti-inflammatory drugs. None of them work. So there is something that unknown that is actually to find out what is.
I can tell you what we found, and it was very different than just blocking inflammation. So in this case, what we saw is that there are two inflammation kinds, two phenotypes. The M1 phenotype, which is pro-inflammatory, anti-tumorigenic, and the M2 like the macrophages, that is anti-inflammatory, pro-tumorigenic. And the microglia and also the astrocytes, there it's called A1 and A2, they change from being anti-tumorigenic, they change their phenotype and polarize to become pro-tumorigenic. They become, their natural function is hijacked by the tumor cells to become allies in a way. Then they start helping them. So originally, if the secretion of P-selectin that we saw, or MCP1, is intended to create inflammation that will attack the tumor cells, then they switch and they become collaborators, like mashtapim. They start helping the tumor cells to grow in mass and recruit the microenvironment, and also recruit those myeloid-derived suppressive cells. So they bring immune cells, but the bad ones, the ones that are suppressive and not the ones that are anti-tumorigenic. And I think this game between the M1 to M2 phenotype is what is different between neurodegenerative diseases and cancer. And we have to look into that and not just blocking inflammation. We have to change their phenotype back to be the good immune cells, to do their work that they were intended to. In this case, we use a P-selectin inhibitor or an MCP1 inhibitor.
Your nanoparticle contains the drugs, they're pro-apoptotic?
No, they are completely, these are...
Mechanism. So what are they doing actually?
It depends which one. The MCP1 recruits the immune cells. It is known to recruit T cells to the site. So, but at the same time, if they recruit also the myeloid-derived suppressor cells and other cells from the immune system that are pro-tumorigenic, then they create a microenvironment that is better for the tumor to grow in mass and spread and become diffusive in the brain, because they become also migratory and diffusive.
But do the cells die?
The cells die at the end, yes. Because what they do,
some mechanism of death.
yeah, the mechanism, it depends which one we're talking about. This, for the MCP1, we see that the mechanism is through NF-kappa B and P65, so we see that that leads at the end to apoptosis. And if we look at the P-selectin, so again, what happens is that the microglia, when we change their phenotype, they start secreting P-selectin ligand, which also affect the NF-kappa B in this case.
So polypharmacology is very important now. It's combination therapy, we know that. And
Yes.
the same, this is the reason I started to develop drugs which are multi-target drugs. One drug doesn't work. You combine the two, get synergies. We see exact, almost identical phenomena in models of Parkinson's disease. And you combine two drugs at lower concentration, the tremendous synergism.
Right. So we're looking at different mechanism of action, different profiles of toxicity, and different resistance mechanisms. Thank you.
Thank you.
Yes?
Referring to that question, did I understand correctly that in the nanoparticle you also included immune therapy?
In the nanoparticle, one modality was to put BRAF and MEK inhibitors together, either tremelimumab and dabrafenib or selumetinib and dabrafenib. This was one nanoparticle. The other had, for prevention, it had MHC1, MHC2 peptides of MART1 or neoantigen together with CpG and the siRNA of PDL1. But this time, the PDL1 is the PDL1 that is expressed on the dendritic cells. So we are not targeting like with the antibody that targets the PDL1 on the tumor cells. In this case, it comes directly to the dendritic cells and silences the PDL1 on the dendritic cells. So that has a much greater effect because it goes to the root of the inhibition from the immune system. So it's not the antibodies that we encapsulated the siRNA.
And why not combine them all, PDL1 with the...
This was the last. The inhibition of metastasis was each one alone, and combining them, and the combination, as Mrs. just said, is synergistic and much more active and less toxic, because it means that we gave 1 milligram per kilo of the BRAF MEK compared to 30 milligram per kilo that is needed for the free drugs.
When will trials begin in humans?
Well, with the P-selectin, we were incredibly lucky because apparently Novartis has an antibody named crizanlizumab that is FDA and EMA approved for sickle cell pain crisis. Nothing to do with cancer, nothing to do with the glioblastoma in specific. So we have already some patients that are treated under compassionate use a month after the paper was published. And now we're working with them to start a protocol to go for a real multi-center clinical trials. Hopefully Rambam will take part in it as well, so yes. Okay, so thank you very much.
Thank you, Rונית, for this fascinating lecture. מדהים. רוצה להזמין את פרופסור יודעים. דוקטור אלברט ופרופסור ביאר להענקת הפרס.
Where's the beef? There was a hat in.
Please.
Thank you. Thank you very much. קורונה, כן.
On the stage we are free.
קורונה free.
You're immune. You've got antibody.
תודה, עירית.
תודה רבה.
תודה רבה לכולכם.